Yeah, good morning, and you're welcome back to the channel. It's always beautiful having you here on the channel. It's beautiful to have time, spend time together discussing science. Today, we are going to look at microbial techniques, and we're going to look at techniques that are used for inoculation. Techniques that are used for inoculation, and this is important. These are basic aspects of our microbiology and the kind of work we do in the laboratory. And it's important that every student of microbiology and student of life sciences in general understands these techniques, when to use them and how to use them. Today we are going to look at three major techniques that we use to inoculate. That means three major techniques that we use to take from our samples and put them on our plates, on our agar plates. Now, as a microbiologist, you want to culture out microorganisms from different samples. There are different kinds of samples. You can have clinical samples, you can have environmental samples, human samples, different types of samples, physics, water, soil, insects, all these different kinds of samples. Now, from these samples, you want to, you know, take from the samples and then put on your culture media so that you can grow out your microorganisms. Basically, there are three techniques that we use. We have the spread plate technique, we have the pour plate technique, and the streaking methods. These are three methods that we use, and three methods that any microbiologist must have a firm grasp of. You must understand what these methods are, how to use these methods, and when to use these methods. Very quickly, we'll go through these three methods. The first one is streaking. Now, when you want to streak, you know that you want to take from your sample, you want to take a little aspect of your sample and then put it on your culture media for growth. Now, for this to happen, you need an inoculating wire loop. There are different types with different sizes depending on what you want to do. And then you have your culture media of interest, EMB, Salmonella, Shigella, Simon Street rate agar, different Muller Hilton, different types of agars for different uses. And so when you have this, your agar is prepared according to your manufacturer's instruction. And then when it's this streaking method, you prepare your agar, you pour it into plates, you allow to cool a little bit, and then you have your sample that has been prepared, whatever sample it can be, fecal matter, urine, soil, even fruits, whatever it is, chicken, whatever it is that you want to culture your organism out from. And then what do? You take your inoculating wire loop, you flame appropriately so that you're trying to kill any microorganisms that may be present on that wire loop. You flame appropriately. After flaming, what do you then do? You put it inside your sample. Your sample will have been prepared. There are processes for preparing the sample depending on the kind of sample it is. Some are prepared with peptone water, some are prepared through filtration, whatever the sample is. But we are just, taking it up from when the sample has been prepared. So you dip in your inoculating wire, and then you look at your petri dish that is carrying your agar. You mentally divide it into four quadrants. You pick one of the quadrants, and then you streak gently with this back and forth movement gently on that quadrant, and then you flame again. When you flame again, you dip inside your sample, choose another quadrant, and then streak. You have this zigzag motion that is done gently in such a way as not to destroy or cause lacerations on the surface of the agar. Rather, very very gently, so that your microorganisms can grow. And that is the streaking method. When you have done that, you close the petri dish and you make sure you are not talking, you are not contaminating your process at that time. And then you incubate over a period of time, 24 hours, 48 hours, 72 hours, depending on what you are looking for. Now, this technique is is the streaking method. And this streaking method is very important. It's being used. A lot of people have used this. A lot of students have learned how to use it. And it's very, very, very important to use. You want to use it for food samples, for environmental samples, for soil samples, for clinical isolates. You want to use them for different kinds of isolates. It's a simple method. It's effective, it's inexpensive. So it's a simple, effective, and inexpensive method. And then its limitations is that you need to really know how to do it. You need to do it under very good aesthetic conditions to ensure that you don't have the contamination of whatever you're doing. So that's the streaking method. The next method is the pour plate method. The pour plate method is also a method that is used. And in the pour plate method, when we've prepared our samples, we take 1 ml of our sample and then we put it into our petri dish, swirl it around, allow it to, flow around. And then after that has been done, we pour 15 to 20 mils of our molten agar. We allow it to cool a little bit so as not to be too hot as to burn off and destroy the microorganisms, but not so cold so that it can pour. And then we pour that amount into the petri dish and then close and allow for growth. In this kind of instance now, you have the microorganisms will be growing majorly within the media or the agar. In the first method, which we talked about, which is the streaking method, you find out that the microorganisms are growing on the surface of the media. In this second method, you find out that the microorganisms will be growing within the media, inside the media. And this is a good method to use to grow anaerobic microorganisms, those microorganisms that do not want oxygen. It's a good method to use to grow microaerophilic organisms that want just very little amount of oxygen. So these are good methods that we can use to grow this kind of organisms. And the pour plate method is good for enumeration. It's good for counting, because if you flip the petri dish over after it has grown, after you've incubated it and it has grown, you can see the different colonies. You can count the different colonies quite easily, and it's very easy to understand. So it helps to provide data. When you say you want to find out colony forming units per gram, you can easily do that. Do your enumeration with this method. And the last method is the spread plate method, which is also similar. And here, what you simply do is that the agar has been prepared in the petri dish. And after the agar has been prepared in the petri dish, you take 1 ml of your sample, whatever sample it is, and then you put it gently, you pipette it gently onto the surface of the agar. And when you've prepared it gently onto the surface of the agar, you look for a spreader, a glass spreader, and then you spread it gently, evenly all over the surface of the agar. That's your sample that you're spreading gently, evenly all over the surface of the agar. And then you close it. And after you have closed it, then you incubate for whatever amount of period and at whatever temperature that you are incubating at, 35, 37, depending on what it is, where the samples came from and what it is that you're trying to grow. So when you do that over that period of time, you're allowed to grow, and then you have your isolates. So these are three major methods that you need to know. Every microbiology student needs to know, needs to understand how to do it, need to practice it many times so that you get dexterity at doing it. If you are going to be isolating microorganisms from whatever sample it is, fruit samples, soil samples, clinical samples, human samples, whatever the samples are, environmental samples, you're going to have to learn how to culture appropriately. And you're going to either be using the spread plate technique or the pour plate technique or the streaking methods. Thank you for listening to this. I hope you enjoyed this. Remember to like our channel, subscribe to our channel. You learn quite a lot as we go along. Have a wonderful day.